welcome to Path Report.
Yeah.
This is our very first episode, and I appreciate
it's an honor to be the first one.
I appreciate you taking your time, Bryce. This is an insight into the world of pathology, and meet your pathologist, so to speak. Pathologists are often thought of as being a stereotypical doctor who is locked away in the basement somewhere, looking at slides, not making it out of the hospital to meet patients. But I want to break that mold.
Definitely.
Show that pathologists are a real member of the care team, do interact with patients, and would like to show the world exactly what we do, how we impact patients' care, and maybe inspire some of the viewers to reach out and meet their pathologist.
Absolutely. I mean, the role is key, and so many patients I don't think really fully grasp it, and so I'm all in.
So as we mentioned earlier, your name is Bryce Olson. Could you tell us a little bit about your background?
Yeah, so I am a technology guy. I've been in technology for a long time. I work at Intel Corporation now, and I am in the health group. Believe it or not, Intel actually has a group that's focused on healthcare. People think of Intel as a chip maker, but we really do a lot more, and we work across many different ecosystem partners. So, folks that are in the health tech delivery ecosystem, along with a lot of different hospitals, payers, life science, pharmaceutical companies, to really help them take our technology and then address some of the bigger business challenges that they have. An area that's very close to my heart is genomics and precision medicine, and we work not only with the large genomic sequencing instrument companies who use our CPUs to really improve the throughput of their instruments and get us to an era now where we're at really low cost, high volume sequencing, but then to take the data off of that and do the bioinformatics, to do the alignment, to do the variant calling, to be able to take on larger studies and to do it quickly using those open source and commercial codes to have those optimized to run on our platforms. And then, we're in the back end really fueling a lot of the clinical decision support systems that are out there too, to take this data and then make something interesting out of it. And so my role in really helping drive that and then tell the stories that emerge from it opened up my own eyes to what was possible for me. Mm-hmm. And so that's kind of my background a little bit.
Great. And Intel is more than just a chip inside your computer. And as we discover about your story, we're really going to find out that precision medicine is kept you alive.
It has, absolutely.
And your interaction in particular with your pathologist who you met was really a turning point for your treatment and your course. But before we dive into all of that, I'd like to talk for a bit about what is precision medicine for some of the listeners out there who may not know or who may have heard of it. It's definitely in the lay press frequently nowadays. And in essence, we'll put up a image here of what precision medicine is, but it's essentially tailoring treatment specifically to the patient's the disease characteristics that are unique to that patient. Precision medicine is not just about cancer, it's about many diseases. Right. But I think the paradigm for it is cancer. A lot of cancers are very amenable to it, and it has really been a success story for a lot of cancers.
Agreed.
In essence, precision medicine for cancer involves doing a biopsy, analyzing that tissue in the pathology lab, the pathologist making a diagnosis of cancer, and then testing that tissue to look for mutations that are unique to that particular cancer for that patient, seeing if there's a weakness that can be targeted and matching that mutation with a drug that may be available to treat that cancer.
Exactly.
So let's start off with how precision medicine has changed your life and sort of before and after, and tell everybody about your disease.
Yeah, so, and you were absolutely right that this whole journey, the whole value chain starts at pathology, and that was exactly the way it worked with me too, because, originally when I got diagnosed, I didn't follow a genomics guided precision medicine path. I was, at one point, when I got diagnosed in early 2014, we thought we might be able to get it all through just standard of care surgery, and when that didn't work, I just, trusted what the oncologists were going to roll out in terms of the standard of care. And we went after it aggressively with both chemotherapy and androgen deprivation therapy. And that bought me some time, but I feel that the chemotherapy was, I don't know, I probably got 9 months out of it, but I paid for it with 6 months of chemo-induced sickness. Neuropathy in my feet that I still feel today, epic mouth sores, just blistering, terrible fatigue, and my cancer started growing again in early 2015, and I really started running out of options. And so that coincidentally was the time when I really started learning about what Intel was up to, because I had transitioned from a general kind of a corporate marketing job that I used to have into the healthcare specific stuff, and that's when I learned all about genomics. And I went to pathology.
And what was
what
your
drove that?
particular cancer?
Oh, sorry. Yeah, I got diagnosed with prostate cancer.
Okay.
So, and it quickly became metastatic prostate cancer, and it was super aggressive.
And that's very unusual for somebody your age.
Yeah, though, as I've increased my network of patients that I've gotten to know, there are more and more younger guys that are, maybe I'm just lucky that I've been able to meet a lot of different people, but I have a lot of friends that are in my age group that have metastatic prostate cancer.
Which is 15 to 20 years younger than the typical demographic.
Exactly.
And
So
you had no risk factors in your
no
family.
risk factors, none. And there's no family history of this disease. And I didn't get the garden variety kind either. I got the really, really aggressive, for folks that follow prostate cancer. I got like 85% of my tumor was consumed with cancer. When they did the biopsy, every single core came back with a Gleason 9.
Okay, that's a high-grade cancer.
Yeah, so it was very aggressive. It spread quickly. My metastatic disease jumped very quickly into bones, and I had to get on top of it. And the pathology, once we were able to really get the genomics stuff going, that's when everything really changed for me. And the conversation was interesting. I learned about this through my work, and OHSU in Oregon, the Knight Cancer Institute, was starting a trial. They had perfected this gene panel that they had been putting a lot of time and effort into. It's called the Gene Trail. It's, I think now it's like 300 plus genes, that are commonly expressed in cancer, and that's what they use as their panel. But they were recruiting, folks for that, and I reached out to the head of pathology that I had just known through my work at Intel, and I said, "Can you sequence me?" And he said, "Actually, we want to recruit you on the trial. We'd like to do it. I hadn't told you this, but I just started looking at your genomics already, and I was going to see if you would be open to being part of this trial."
It was very coincidental.
Yeah, it was super coincidental. But what really changed the course for me is he said, this is what he said. He said, Dr. Chris Corliss, actually at OHSU, runs the Knight Cancer Institute for all the genomics and pathology. He said, "I think we have something that could be clinically significant, but I'm not really supposed to talk to you until I've had a chance to talk to your oncologist, and I haven't done that. This is literally coming off of the analytics has just been run, and that's why I hadn't reached out to you to consent you in and everything, but we did it as part of a research thing, and would you want to be part of this study?" Or it was something like that. Those details are really not that important, but at the end of the day.
But the fact that you sought him out,
Yeah.
and he had already started to work on your tumor and generate some data.
Yeah, and when he told me that he thinks that there could be something clinically significant, it's in the PI3K path. You have a P53 mutation, but you also have a PI3K you seem to have your PI3K pathway seems to be mutant, and you have P10 loss, which, you know, if your PI3K pathway is mutant and you also have P10 loss, it's almost like your body put a brick on the accelerator and then drain the brake fluid. I mean, it's really, there's no tumor suppressor gene that's doing its job in that pathway.
And I think we have a graphic that we're
perfect.
show the pathway that PI3K is integral to cell division, growth, and it's, as you mentioned, an accelerator.
Yeah.
And the P10 acts as a regulator or a brake.
Exactly.
And with your particular cancer, you had a sort of double hit.
A double hit.
Both were mutated.
Yes, and the way that you articulated and the way that I was trying to articulate it, I couldn't do that when he first told me. When he started mumbling off these, you know, these 4-letter acronyms, 3-letter acronyms, I had to stop him, and I said, look, you need to invest a little bit more time in me. I'm asking you to invest a little bit more time in me to get me up to speed on this, to help me understand what this really means. Because if you don't, and you just talk to the oncologist, and then I have a meeting with him, my guess is that we're not going to follow whatever insight you just found.
Something may be lost in translation.
Now, either something's going to be lost in translation, or he's going to look at this and say, I don't know, this is experimental, it's not standard of care, I don't know the evidence behind this to put my patient on, you know, based on the fact that you see some hyperactivity along this pathway and that might be driving the cancer, to take my patient off of anything that I would recommend as part of a standard of care and put him on this is probably something that I'm not going to do. That's my guess of what his philosophy would have been on this. And so, again, this was 2015, early 2015, you know. Yeah, and so I just said, you have to, Corliss, please, you know, educate me on what this means. And he did, and I felt empowered.
That's fantastic.
Yeah.
I definitely appreciate it when patients approach me and feel that that's the best care, when a patient becomes empowered, wants to learn about their disease.
Yeah, and I agree. And once I, in some ways, like you hear a lot of people talk about in cancer that you get empowered as a patient when you get diagnosed, and you get empowered when you get a recurrence. But for me, this was a molecular diagnosis, a new diagnosis, and it was personalized to me. So my empowerment meter went really high. And
this is personalized medicine.
Yeah, and so I really wanted to take this to the periphery, because I already know, you know, I got diagnosed at 44 years old. I know what the median survival stats are for a guy whose cancer is metastatic to the bone, progressed on chemo. I don't like that outcome. And I'm not, you know, I don't want to just eke out another couple years. I want to go outside the lines, because I got a 9-year-old that I need to get through high school at least, right? So for me, it was really critical to try something new. And I was willing to take the risk. There was somebody today at this PMC, this Personalized Medicine Coalition conference today, had shown some data and said something like, hopeful, safety, like hope, like taking informed hope or something like that is much better than a safe bet. And I agree. I think for someone like me, I'm willing to roll that dice and take a little bit of a risk if there's some data behind it. Absolutely.
And I think that's something we're going to talk about a little bit later on, is the value of precision medicine.
Yeah.
There's some exciting data coming out that you're aware of that really shows that the investment up front does offer survival advantages, value downstream, and we'll talk more about that. So you really did a lot of self-education along your journey, and your pathologist sort of helped you, guided you along that way.
Yeah.
So knowing the particular characteristics of your tumor, then how did your treatment change? Yeah,
so it's a great question. So there were, after I progressed off of chemo, if I wouldn't have done this, most likely I would have fallen into the remaining standard of care treatments for metastatic prostate cancer, which are a lot of secondary hormonal treatment, you know, things that kind of mess with the androgen receptor signaling inhibition and things like that. And I didn't want to go there for a variety of reasons. One is I'm a little freaked out, right or wrong. I'm a little freaked out about when you inhibit the androgen receptor that heavy and that hard, the resistance mechanisms in potentially changing that cancer from an adenocarcinoma to take on neuroendocrine features. There's weirdness about that we don't really know. There's a lot of fatigue and even cognitive complaints that I hear from patients.
So a lot of side effects.
Yeah.
Because that's still general chemotherapy.
Yeah.
As you had previously. And
Yeah.
suffered from a lot of the side effects.
Yeah, and then again, I know what the data is. Sure, you get a little bit of a bump in median survival, progression-free survival, overall survival, but it's not what I was looking for. So I took that data that he gave me, and I went out and said, okay, well if I have hyperactivity in the PI3K mTOR pathway, then I need a PI3K mTOR inhibitor. And so I found a phase 1 drug that was being tested at Cedars-Sinai in LA, and I used that data, and it was funny, I called them up and I said, I want in, and they said, I'm sorry, there's no space available. Just all these advanced cancer patients in the LA area are rolling into these trials, I'm sorry. And I said, yeah, but I got sequencing data. And she said, you have what? I said, I have genomic sequencing data. I know I'm a perfect molecular match to the drug you're trying to test.
Right.
And she said, wow, no one's ever called us and told us that before. Can you come in? We think it's interesting to build potentially a mutant PI3K cohort. And though the drug company didn't necessarily want to go there in phase 1, I'm curious. The principal investigator said, I'm curious and I want to do this. Great lady, Dr. Monica Mita at Cedars-Sinai. And so she enrolled me.
And you must have learned in your research that this is a rare mutation in prostate cancer. There's some data in breast, I think. There's a number of trials,
Yeah.
but prostate.
I was the only prostate guy. I think I was the only prostate guy in this trial, this phase 1 trial that they were doing. But what's really interesting is the drug ultimately failed. A lot of phase 1 drugs do fail, cardiotox, but I didn't have that. And for me, it kept my disease stable. I didn't get for 2 years. And the drug, I think after maybe about a year and a few months, they stopped it. The FDA came in because of the cardiotox issues and canceled the drug. But I was able to show that not only did I stay progression free, but I think 1 or 2 of the Mets didn't even show up on imaging anymore. It was like,
you actually had some regression.
Yeah,
of the disease.
and I used that data, and my principal investigator, Dr. Monica Mita, used that data to solicit support from Cedars-Sinai and the FDA to give me single patient IND status for the remaining drug supply in that phase 1 drug. So I was on it for 2 years, which is an amazing, exceptional response, I think, when nothing else worked, And then coincidentally, in the last month that I was on before the drug supply totally went away, I did start to get a biochemical recurrence. So
Your PSA.
yeah, my PSA started to go up. So, progression-free survival of 2 months. Biochemically, we saw a recurrence. It took several months after that to start seeing it radiographically in some vertebrae that I have, but I bought a lot of time.
So it was, even though the drug failed, personally for you, it was a success.
Totally, yeah, complete success. And it's what made me a precision medicine advocate and evangelist, because I know what the outcome would have been otherwise. And I'm here today talking to you because, and not only here today talking to you, but I have energy and I'm healthy and I'm working and I'm productive and I'm generating an income and paying taxes and all these things that, somebody who's been battling stage 4 aggressive metastatic cancer since 2014 may not be doing.
Right. This is value.
Yeah, exactly.
So this brings you up to 2016? Or?
No. I started that drug in March of 2015, and in March of 2017, I started getting the recurrence.
So earlier this year.
Yes.
And you learned some additional features about your tumor.
Yeah.
So was additional sequencing
Yeah.
done, a repeat biopsy then?
New biopsy.
And that's another important concept of precision medicine, is that tumors evolve.
Exactly.
They change, they can evade treatment, and one of the weaknesses of precision medicine is that tumors often develop resistance.
Yes.
And we have to come up with a second line and a third line.
Exactly.
So it sounds like you were in that category looking for
That's exactly right. So I know that my cancer had somehow, towards the tail end of that PI3K/mTOR inhibitor that I was on, something broke loose. Something new is causing my cancer to grow in spite of using that drug. So I was very curious. Okay, so what is it? What's fueling my disease? What's new?
And a new metastasis was biopsied, or?
Yeah, so that's what we did. We said, okay, well, the only way we're going to really determine this is if we go after a new met. And so I had one in my thoracic vertebrae, my T5, and it was kind of posterior and just under the skin, and so it wasn't that difficult to get to. And so I went to UCSD, who has a really good reputation of taking DNA off a bone, like doing biopsies, extracting DNA off the bone successfully, and doing a number of things, including growing organoids in the labs and xenografting it into mice.
Right. And I know as a pathologist, whenever I hear bone lesion and molecular, they usually are not compatible.
Exactly.
The decalcification
process
renders the tissue often useless for molecular profiling. So that's terrific that you had a novel way to get the information.
Yeah, and it was really interesting. Like Dr. Christina Jameson at UCSD we got 4 bone fragments. 2 of them she put into cryo vials and saved them. One of them, at least one, was sent over to TGen, the Translation Genomic Research Institute in Arizona, who volunteered to step up and do DNA and RNA sequencing of that. And then my primary lesion data, the exome data, was sent from OHSU to TGen so they could do the comparison of primary and metastatic. In parallel, raw data off of that, from that sequencing effort, was sent over to UCSD, and Dr. Pablo Tamayo and his team are doing a number of analytics exercises on that in parallel too. So, and some of that data is coming in like right now, like real time. Right. Yeah.
And what's have you learned new about how your tumor may have changed?
Well, a couple things. So they had a new hire over there at UCSD, Ludmil Alexandrov, I think is how you pronounce his name. Real smart guy that studies somatic mutations and looks for passenger mutations and things like that. And he looked at my primary disease and then also the metastatic disease, and saw that, yes, certain percentage of those molecular signatures, the passenger mutations, are associated with aging and some with APOBEC, but he saw 60% of them were associated with HR, how do you pronounce that?
Homologous recombination.
Yes, and yet I don't have any BRCA mutations. I'm not a BRCA1 or BRCA2 DNA damage repair mutation. I don't have that. However, there's some new data out now that's looking at, well, you don't necessarily have to be BRCA1, BRCA2 mutant to potentially respond to a PARP inhibitor. It's possible that if you have a lot of HR, if your mutational signature, if you have a large percentage of HR, you might be a good candidate for something like that. So that was new and interesting. I would have never thought about that.
And I think we have a graphic here that we can put up that explains about DNA repair, the theory, concept behind it, and PARP inhibitors.
Yeah.
So, these PARP inhibitors take advantage of a tumor's weakness by preventing the repair of strand breaks of DNA. And typically, people will have 2 pathways to which repair DNA, through the PARP pathway, but also the homologous recombination, which repairs double strand breaks. And in patients who have a deficiency in one pathway, we may be able to inhibit the others to cripple the tumor so that it can't repair its DNA. And this is what we term synthetic lethality, which is something that is gaining more and more press and acceptance as a new line of targeting tumors.
Yeah,
so
exactly.
That sounds like you may be a candidate for this.
Potentially. And actually, it's interesting you mentioned about the synthetic lethality, because one of the collaborators at UCSD is also Dr. Don Durden, He's got the Durden lab, and he studies a lot of PI3K related illnesses and a lot of things with children. He's part of Ratty's Children's. But he is looking at dual inhibitors and triple inhibitors, so like PI3K, BRD4, other things, other resistance mechanisms that are commonly associated with PI3K inhibition, and how do you potentially get synthetic lethality with, potentially, one molecule that could hit multiple targets. And so he's involved in looking at my stuff too, and trying to, ultimately take the genomics that come out of this and infer what would be a, exploratory treatment 1, 2, and 3, or maybe a combination of these things, and then potentially in the wet lab test them against organoids to determine, whether that's a great model or not. It's to be determined, but at least it would give some preclinical data. And so so we're looking at that. The other thing that the UCSD team is doing is they're plotting. This is kind of interesting. They're taking my genomic data, plotting it against something that Dr. Pablo Tamayo worked on, which is called OncoGPS. And so they're looking at different cellular, like oncogenic pathways, and then putting those into states. And then kind of plotting, okay, Bryce lives on this state. It's like, okay, where's my pin? Pin drops here, relative to all these other metastatic cancer patients that are part of the Stand Up to Cancer metastatic prostate cancer data set that Ellie Van Allen, Dana-Farber, and those guys contributed to. And looking at where I sit relative to them. And it was funny, today I was at Dana-Farber talking to Dr. Ellie Van Allen, and I told him, I said, you know what? It looks like there's 10 or so patients out there in the database that look like they're nearby neighbors to me from a molecular perspective. There's one guy that looks really kind of similar to me. Can we look at the outcome, the treatment and outcome data of this guy and see what he did? Because it could be informative. And it turns out he's some guy who was treated at Sloan Kettering, and it's interesting what they're able to kind of look at.
Right. So we've taken precision medicine, tailored treatment to a personal level, but then we've collected patients on a population level and backed up from a larger view to look and see what similarities there are, what can we learn from the collective data.
Exactly.
And I think that's something we can talk about in a few minutes about where we could use some more work.
Yeah.
I think. So you also learned something else about your tumor. It had some characteristics of what we call epithelial mesenchymal transition.
Yeah.
And we have a graphic of what that is, but essentially that's cells have a degree of plasticity. They can change phenotypes. They can change their characteristic from being a epithelial cell to a connective tissue type cell. And in so doing, this is a normal process that we use for wound healing, growth, development, but tumors take advantage of it by utilizing it to metastasize and spread.
Yes, that's exactly right. And especially, as a disease moves from a primary epithelial, cell to becoming something that's actually attached to bone. And you're right, it goes through that kind of a transition. And I don't think I had, at least the amplification that they showed on my primary relative, it's different. The EMT is definitely pulling harder in the metastatic. They even see potentially in the metastatic lesion, it's still early, they just literally got the data a couple days ago, but I might have a little bit of MYCNIS, MYC.
Right, which is another oncogene that drives some other tumors, often lymphomas, but we're discovering more and more tumors may express and utilize this oncogene.
Yeah, exactly.
So you're really on the cutting edge of precision medicine and our understanding of cancer, our approach to it, and the therapeutics.
Yeah.
You're right there.
Well, and I feel like, by the way, there's also something interesting to what you just said about that too, and I feel like I've kind of had to from a survival perspective, partially because I think up until now, yes, Corliss and his team. Jamie Keck is someone who works for him, by the way. He became a close personal friend, and I used to, bounce stuff off with her over coffee and brunch and stuff too, because, I mean, she loved the fact that I was a patient who was kind of educated in this, and, her mom actually went through stage 4 colio, like the bile duct.
Right.
And was diagnosed, genomically profiled, got onto target treatment. She survived a lot longer than probably normally would have. But I just tell you this because the pathology team, they love when you get a patient that understands this and they can consult with and bounce things off of. And for me, it's been tremendous to have that, because the unfortunate state of affairs today is a lot of oncologists, they're just not, again, they're doing the evidence-based things that they know that is part of their treatment plan. And for me, I want to just kind of explore the periphery, and it's been pathology, it's been the right partner for me on this. So, and I'll continue to do that. But, I know the point, sorry, I was circling around to, I did finally find a medical oncologist at UCSD, Dr. Rana McKay, who's a young, aggressive, energetic. She wants to take a lot of this genomics and exploratory stuff, potentially combine it with standards. But I feel like she wants to explore the space with me, and I think for the first time, I feel that I have a doctor who I can kind of like maybe relax a little bit, that I don't have to always stay on top of everything, that she is on top of all this stuff and will guide me. And that sense of relief, it's exhausting to kind of be guiding your own care.
That was my next question, was out of this, what have you learned? Because you've been a really strong advocate for your treatment path and seeking out the newest, the best.
Yeah.
But what about patients who don't have that energy or don't have the resources?
Yeah.
How do we help them?
Yeah, no, it's a great question. It's a great question. I mean, I think in some ways, you don't have to, by the way, it might sound like, wow, he kind of knows what he's talking about, but I was a business major. I was a marketing guy. I didn't even like science in high school, and I didn't study it in college. And yet, I got up to speed on this stuff. So it's not, to be at the level of how to articulate and understand this at my level, it may sound like it's pretty high, but it's not. The learning curve wasn't as bad as you think. So that's number one.
Hopefully some of the people who watch this video will take that message and see that it isn't that hard to do what you did.
Exactly. And then the other thing, or a couple things, is, I think if you're an advanced cancer patient and you're running out of options and you don't feel like there's hardly anything left in the standard of care toolkit to help you, if you can just understand that genomic sequencing can potentially open the door to give you some new answers. It may not lead to anything, but it could open the door. And so ask for it, because if you don't ask for it today, it's not standard today. So you have to go into your doctor's office and say, I want to be molecularly profiled. I want genomic sequencing. I want to know what's fueling my disease at a molecular level. So give me the answer. And they're either going to be a big academic cancer center who has the ability to do it themselves, or they don't, and they have the ability to take your tumor and outsource it to a Foundation Medicine or somebody like that, and you can get new insights back. Right. It's as simple as that. And I did a couple of other things. I started a little kind of a movement called FACS, Fighting Advanced Cancer Through Songs, which is kind of just meant to use music as a way to raise awareness for a new way to fight cancer that people might not be. If you've got a mom or dad who's dying from cancer, and you're the son or the daughter, and you don't know much about this, but you kind of like music, and you got roped in, and you heard about a new way to fight cancer through songs, fine, great. I was able to expand the knowledge base of people out there. And
right,
so,
and we've got a short video of the FACS movement, and we'll roll that here.
Music gave me a way to cope with a terminal cancer diagnosis that was really unresponsive to the standard of care. Genomic sequencing gave me targeted therapy and precision medicine that finally stopped my cancer from growing and gave me a life that I didn't think I was going to get to live. Demand genomic sequencing because genomic sequencing will open up the door to a new way to fight cancer.
And that's when I met you
Yeah.
earlier this summer. You walked onto the stage with a Sequence Me shirt,
Yeah.
which I thought was really interesting, with everybody else sitting in suits.
Yeah.
And
Yeah, thanks.
tell me about that shirt. And it
Yeah.
goes hand in hand with your passion, your advocacy
Yeah.
for learning genomics.
Yeah, absolutely. So for me, as part of this Fighting Advanced Cancer Through Songs, I wanted to try to figure out something that would be short and succinct and interesting enough that people might question, well, what does that mean? What is that? I want to learn more. The part of Fighting Advanced Cancer Through Songs was songwriting. For me, was very therapeutic. Music can be very therapeutic, and as a way to just connect into feelings of sadness and frustration and anger and things like that. Well, for me, songwriting on top of it was how I expressed those things. I wrote a bunch of songs. I started talking to people once I learned about this genomics, that I said, look, I want to take these songs and do something meaningful with it, turn it into more of a movement. And I asked all these Portland artists in Portland, Oregon, musicians and singers, like, would you be interested in being part of this? And they all jumped in and said yes.
And some of them are very famous.
Yeah, some of them are really famous. Martha Davis from the Motels, for example, Jenny Conley from the Decemberists, just to name a couple.
Mm-hmm.
And so we released an album, and one of the musicians, her name is Michelle DeCourcy, who is also a very well-known fashion designer. She designed, oh my gosh, I'm going to blank on her name, but Norah Jones, when she won the 6 Grammys and she came out with a dress. That dress was designed by Michelle DeCourcy, who helped me design the Sequence Me shirt, and so she gave me that, and she's on the album as well. And the album, again, it was put out there to raise awareness for a new way to fight cancer, to drive people to the website on Facebook to learn more. And we're also raising funds for precision oncology. Actually, this week on Friday, I'm part of my fundraising effort was on a crowdfunding site called consano.org, and the CEO of that, Molly Lindquist, and myself are meeting with Gordon Mills, who's OHSU's new hire out of MD Anderson to run precision oncology, and we're giving him a $40,000 check on Friday that we raised.
Wow, that's terrific.
Yeah, because he's going to go drive a bunch of precision oncology trials, and we want to get more patients into those things.
Right, and enroll more patients so we can learn more.
Yeah.
How can listeners at home help out? Go to your website?
So we do have a www.faxmovement.com page. I think more the interaction and engagement is probably occurring on Facebook. So on Facebook we have a fax, Fighting Advanced Cancer Through Song page.
And they can follow you on Twitter too?
Yeah, Twitter, Bryce Olson, at Bryce Olson. And I think, new things are cropping up with me, like I mentioned, that I'm going to get out there. I am also going to highlight, findings that I heard from today and other precision medicine conferences that I go to. And ultimately, what would be kind of cool, I think, I just haven't had the time, is if the fax page is becoming this place where patients and caregivers who are trying to fight cancer with a new way, if this is becoming, one congregation area for them, it'd be interesting to start bringing them solutions, and exposing them, not only just education, but actually bring them solutions from the pharmaceutical companies, from the hospitals, from the commercial diagnostic test companies, things like that.
And even your work with Intel.
Yeah.
Help with some of this
exactly.
data management.
Yeah.
Well, I really want to thank you for your time today.
My pleasure.
It's been a real pleasure to hear your story, to be honored to tell our listeners about it, and I think importantly is to be your own advocate.
Yeah.
And one of the key points in your journey was to seek out your pathologist.
Yes.
And I encourage listeners to find out who their pathologist is.
Absolutely. The path to precision medicine for me started with pathology, and I wouldn't be on this path if it wasn't for pathology.
So definitely check Bryce out on Facebook, on Twitter, the Fax Movement. You can go to pathreport.org for more information, and we definitely welcome viewer feedback, suggestions for future episodes.
Yeah, awesome. Well, thank you. It was a pleasure.
Thank you.